I mean, well, we can wait till people come on.
Huh?
That's okay.
No, I just hit record. So, because I was just gonna do the practice exam, and so there's already kind of an answer key for that. So it wouldn't really make sense to post. And I lost you.
Okay, these headsets is terrible.
Yeah. Yeah. So I'm gonna record.
Okay, all right. Okay. Is that posted in the announcements? That right there?
Yes, this was posted. Let me see if I can pull up another tab actually, so you don't see my screen. Not that day. April 2nd. April 4th. It was posted on April 4th.
April 4th.
Yep. So I know it's 6, but I'm gonna give maybe 1 or 2 minutes and then I will get started since it is recorded. So
Okay.
So for this one, I know is it a cyclo or is it the benzo?
this one is
Benzoacetic. It's a cycle,
yep.
and it's 1, 2, 3 on this one, and 1, 2. Did I count that right? 1, 2, 3. Is it 3 carbons on there and then 2 on the other one?
1, 2, 3. Yes.
And that would be so. That will make it let me think about that.
Yeah, so we'll go ahead and get started. How I was going to do this is I was just going to go through the practice exam and do each problem and talk them out, and let you guys ask questions. And then if we had time at the end, I'll just take open-ended questions about anything, any OWL homeworks or any other topics from the exam. So this is the practice exam that I posted on an announcement on April 4th. So looking at question 1, Taylor just talked about trying to identify if this was an alkane that we use the prefix cyclo for, or if this was a benzo something. And this is an alkane which we use the prefix benzo for, or I'm sorry, cyclo for, because it has all single bonds and no double bonds within the ring structure. So we know it's an alkane because it's all single bonds. So it's going to end in ane. We have to figure out how many carbons are in our parent chain. So our parent chain, if there's a ring and it's all alkanes, then our parent chain is always the ring. So this is a 6-membered ring. So it takes on the prefix hex. So right now we know that it's a cyclo because it's a ring, hex because it's a 6-membered ring, and then ane because it's all single bonds. And so then we just have to name our constituents or our branches. So numbering, we want to put the lowest, we want to put the branches the lowest numbers possible. So that would either be 1, 2, 3, 4, 5, or maybe going this way, 1, 2, 3, 4, 5. And so what it comes down to is putting them in alphabetical order. If there's no other like functional group taking precedent for that first carbon, then we would just put our branches in alphabetical order. So this is a 2-carbon branch, so it's an ethyl. And then this is a 3-carbon branch, a propyl. So we're looking at ethyl being the lower in the alphabet. So we'd want to number with that's the 1 on the ethyl, 2 on the propyl, and then 3, 4. Oh, sorry, this would be 3, then 4, 5, 6. So then it's a matter of putting those in the front. So 1 ethyl, 2 propyl, cyclohexane. Any questions about that one?
There's no space in between that, is it?
No.
It's like, okay.
So there's no space, but good news for you. This is multiple choice, and so
Okay.
it's just a matter, and they don't test you on the format. So like you don't have to, you only really had to grill yourself about that for owls.
Okay.
Okay. So Angelica, I know you just joined, but I am recording this. So and we've only done the first one so far. So any other questions about one, or I'll move on? All right, I'm gonna move on. All right, so drawing the structure. So in the practice exam, I had you draw them. Again, you'll be given A, B, C, D of structures. That was a lot for me to try to make. So working backwards, we know it's an alkane. So all single bonds, and then there's 4 carbons in the parent chain. So I'm going to go ahead and draw 1, 2, 3, 4. So now we have our 4-membered parent chain. And so now it's a matter of,
Professor Daniels.
yeah.
I'm sorry. So it's 4 because of the butane, right?
Yes. Because of the
Okay.
yes. Thank you.
And it's an alkane because what did you say?
Because it ends in
Because
A.
it's on in A.
Yep.
Okay, I'm making sure I got that. Okay,
Yeah.
okay.
So if I number right to left, I would put my ethyl group on the 2nd carbon, which is a 2-carbon branch. So 1, 2, and my methyl group on the 3rd right here. Now, because we were, oh, there's one more person. Okay. Because we were drawing it, we got to choose, but remember this is multiple choice. And so it's possible that, you know, if you don't see this answer choice at first, but you're confident that's an answer choice, try numbering it the opposite way and see if that is available. I haven't gone through the exam and looked for that specifically. How they number them, like if it's often left to right but so both of these are basically the mirror images of each other. So they're both correct. Any questions about this one? Okay. So the third one, which molecule has the highest boiling point? So I left this one on the exam because they had the correct answer. There is another like boiling point question in which I removed because they didn't have the correct answer even listed. So it was kind of, yeah. But so this one, the highest boiling point, they all end in ane, meaning they're all alkanes. Functional group. So now we have to ask ourselves, well, how do we compare boiling point or basically properties against just alkanes? And so then we look for which one has the highest number of carbons if we're comparing all alkanes. So methane has 1 carbon, propane 3, hexane 6, octane 8. So which one has the highest boiling point?
Octane.
Octane. Yep.
Octane.
Yep. And then I had two more people who just joined. So I'm recording this, but we're only on question 3 and we're just going through it. I'm going through it as if I'm, taking the test, and then you guys are allowed to ask questions as we go through each one. Okay, so any questions on 3?
Methane. Yeah.
All right. I'm gonna go ahead and clear this. All right. You have, I think, 2 questions that ask you to identify functional groups on the exam. So one of them, it just shows you a functional group with no thing attached to it and wants you to identify it. And then another one, it shows you a molecule, so with stuff attached to it, and then ask you to identify the functional group in that molecule. So what functional group is present here? It's a carbon. Yeah, carbon double bonded to an oxygen, bonded to an H. All right. Just because I didn't give you the exact one that we're going to see on the exam. Let's go through the other answer choices and determine. So what would a ketone look like?
The ketone had a double bond in the middle.
Yep. And then it'd be sandwiched by the rest of the molecule, basically. So there'd be other carbons here. What about an alcohol?
Professor Dennis,
Yeah.
I'm sorry. I just got a question.
Yeah.
I see you see the picture right there shows you got the C's for the carbon.
Yeah.
But I'm looking at the answer key. Like, doesn't matter what's the R for, it just don't matter.
Oh,
It's just the same as
Yeah. So R just means basically whatever else is in the molecule. So
okay.
Yeah, does that make sense? Yeah.
Oh. Yeah, I was just
There
wondering.
like other carbons.
Other carbons, this side could have 3 carbons and then 4 carbons, or this side could have 1 carbon, but as long as it's sandwiched between a double bond sandwiched between 2 carbons, then it's ketone.
Right. So the R basically, I think you were saying something in class like resting the rest of it or something like that.
Yeah, just the rest of the molecule, whatever it is.
Okay. And a ketone is the double bond carbon in the middle.
Yep.
Basically. Okay.
All
right.
What about an alcohol?
That's an
Bonded to an
OH.
OH.
Yep. And then carboxylic acid. Running out of room.
Carbon bonded to an OH and a double bonded O.
Yeah.
Double bonded O and the OH. Okay.
All right. Just because you guys are all awesome and you came to the exam, what would it be We didn't stress this one too much. Actually, give me a second. Gotta make sure I drew
Okay.
that.
Let me put it on mute because my kids singing in the tub. I don't want nobody they just singing.
The example that you just put on, is that when they use like the end because we don't know exactly which branch is on?
Not necessarily. So this example is one that I drew after, I think I added to our functional group chart, but we didn't really like stress it too much. So this is a propyl group, right? So if this was hanging off our main chain, it's 3 carbons is a propyl group. But when it's specifically attached to this middle carbon here, it's called an isopropyl group.
Only in the middle when it's attached, only in the middle?
Yep.
It's an isopropyl.
Yep.
Okay.
And don't think too hard about it. You're only going to see that on the question that talks about identifying functional groups.
Hold on, I don't see it.
You don't see what?
No, all I see is you on my screen.
Oh, I don't know how to help you there because
You have to move your the faces, move the top of it to the top of your screen. Then it'll show you the rest of what she just wrote.
thank you. Did that work?
For some reason it's tripping. There we go. I see the screen now. There we go.
Okay.
All right.
You had me panicked because I was like, I don't know how to fix that problem. You think you're good at technology and then you teach a class and you're like, wait, I don't know how to fix some of these problems. Okay. Any questions about this one?
Wait, so the isopropyl is the CH3.
Yes.
CH3 with CH3.
Yes.
Okay. With to the left of that is the rest. It's basically our
Yeah. That's just whatever else you want to attach that to. All right. I'm going to go ahead and clear this. All right. Question 5, naming. So first of all, what kind of suffix am I going to use? What functional group is present?
YNE.
YNE? Any other?
ENE.
ENE. There we go. So it's an
Oh shit! My vision blurry. That looked like a triple bond.
yeah. So it's alkene. So ENE. Do I want to number it right to left or left to right?
Left to right. The bottom one.
The bottom one. Okay. So, yeah, so that puts, when we have a functional group present, we want to put, that takes precedent over trying to make the branches low. So we want to put that double bond as low as we can. So putting it from 2 to 3 is more advantageous than 3 to 4. So we have a 5 carbon, so pent. So I'm going to start to put this together. Pentene. I need to tell my audience where this double bond's at. So it expands from 2 to 3. We just have to tell the audience that the lowest of the carbons that it stands. So
it's 3-methyl.
2 pentene, yes. And then getting to the methyl group. So there's a methyl group off of carbon 3. So like Taylor just said, then we add 3 methyl, and I'm sorry, I'm running out of room here. 3 methyl 2 pentene. All right. Any questions about that one?
Yeah. I'm trying to solve it in my head. I see it, but I'm trying. Okay, the two. One.
The equal sign just means that it's a double bonded, correct?
Yes, yep.
So that's that 2 pentene. That's where the double bond is at on the. No,
Yeah.
it's on a 3. It's the
It's
double
5
bond's on the
C's,
3.
5 carbons.
So we ended up
Yeah, I see the
we
five
ended up
carbons.
going with the second naming, and it does span on 3, but it goes from 2 to 3. And so when we tell the audience where it's at, we just need to tell them the lower of the 2 carbons.
Okay.
All right.
Okay.
Any more questions? All right. So looking at 6 here. So we still have an alkene because it ends in -ene. Right? So bute means it's going to be a 4 carbon parent chain. So I'm going to go ahead and draw 4 carbons. 1, 2, 3, 4. Oh, yeah. 1, 2, 3, 4. And then it tells me that that double bond spans from 2 to 3. But now I need to address the fact that it says cis. So as I have it drawn now, it's actually trans because I've put the 2 groups opposite of each other on the double bond. This would be where they're on the same side. So this would be 1, 2, 3, 4. So the correct answer is the one on the right.
I just kind of want to get it done today. So even if it's right, I'm not gonna-
So even if
Don't rest that yet, Professor Dennis. I'm sorry. I'm just trying to draw it myself. Don't give me a second.
you're fine.
What made the second one, it look like it's still on the same side? What made that a cis? The cis means that it's on the same side, right?
Yep, cis means it's on the same side. So the heavy groups, which in this case are the carbon, the carbons here,
Mhm.
they're on the same side of the double bond. Whereas when they were on opposite sides here in the trans version, it's kind of hard, like cis and trans is kind of, I don't know, it's difficult.
And then
We did it in the lab, it was easy to identify, but I just don't see how they identifying it now.
Okay, so 1 and 4 are on
That's what I said. I'm like, I don't know why, because I'm sorry to interrupt, Professor
yeah,
Dennis.
yeah.
I don't know why it's looking like tricky to me now because I was catching it and then I'm looking at it like, what am I missing now?
Yes.
But I'm
So
a catch.
did you know that there's a double bond at 2 because it says cis 2 and then because it's an alkene?
yes.
Okay. So could you essentially put it the other, like label it the other way?
Yes, so I'm gonna clear, but don't freak out because I just want to start like have some more space. So here was, so you said the other way, and I think, do you mean this versus?
I mean like labeling it
oh, like if I did 1, 2,
yeah.
3, 4.
Yeah, like it doesn't matter left to right with this one.
It doesn't matter. Nope.
Okay, so the way that you just labeled it now, does that make it on the opposite side? Is what you're saying from the bond and the numbers, the actual carbon?
So the labeling doesn't make it on the opposite side. What's the opposite its side here is these two. So like, oops. So here's my heavy carbon groups.
Mhm.
The double bond is my axis here, and they're on the same side of the axis. Whereas when I had trans
Mm.
and the double bond was my axis, they were on
Oh,
opposite
got it.
sides.
Got it. Okay.
That makes sense. Okay.
Thank you.
So trans is opposite and cis is the same side. Okay.
So in this example, cis will be this first one and then trans will be the second one.
Yes, yes. So
Got
this
it.
is the correct answer to the question, but you know, just if for reference, that's what trans looks like. All right. Moving on. This at the top. Okay. So reactions, right? So this is what I'm imagine you guys are gonna struggle with the most because there's a lot of components. And unfortunately, they go from name to name. So you have to know what, not only is it testing you on, you know how to draw that structure, you know how to do the reaction and know what it creates. So first of all, hexene, alkene functional group, reacts with water under catalytic sulfur acid to form what compound? All right, so this is exactly the wording in the exam. I just changed what molecule we're working with. So if we see something alkene reacts with water and then all this other junk, this is the reaction that we're doing. So when we do that, this is when we had like alkene bond. Let's say it looks like this. We break water into H and OH. We add H to one side and OH to another, and then we break the double bond, right?
Propane.
Yep. So if we started with hexene here, 1, 2, 3, 4, 5, 6. It doesn't tell us what number that double bond is at, so we're just going to assume it goes from 1 to 2. So when we do this reaction, hydrogen goes to one of the carbons and OH goes to the other carbon, and we break that double bond. The rest of the molecule stays the same. So it would end up looking like this. And there's a hydrogen here, but I don't necessarily need to draw it out because all of hydrogens are implied in our structures. Now, I said there was 2 correct answers. I wrote this before we had talked about the Markovits rule because because I didn't realize that was going to matter. But it does matter. You're going to have an answer choice that says 1 hexanol or even 2 hexanol or 3. But we want to add the heavier group, which is the OH group, to the first carbon, to the less branched carbon, which happens to be 1. So now we have an OH as our functional group in a 6 carbon structure. So we've changed the ending to enol, but we still have hex. So hexanol, and we need to tell our audience where that OH group is coming off of. And it's coming off of the first carbon. So the correct answer choice is A. Questions here?
Yeah, Kayla, so you said that we had to break the double bond, right?
Yes.
So
Yep.
when we broke the double bond, where did our hydrogen go? I know our OH is always going to be our
Yeah.
first one, but
So the H,
where did that hydrogen go? Where was it implied to go?
it went on 2, so it went on the other side. So 1 part goes here, 1 part goes here, break the double bond.
Okay.
And then I left it off of the final drawing because it doesn't necessarily participate in the naming of the molecule.
Okay, because it's just implied, right?
Yep,
It's one of those
yep.
things that's just implied. Okay.
All right. So another alkene reaction.
Professor Daniels, I'm sorry, I got a question.
Yeah.
The one we working on right now.
Yeah.
You said that you didn't know where the double bond going, so you said the lowest carbon would be between 1 and 2. That's why you put it right there.
Correct.
Okay. Okay.
All right. Okay, so 8 is very similar, only we're changing what we're reacting with. So it's an alkene reacting with HBr. So, I can't remember if HBr is exactly what's on your exam, but it could be HBr, it could be HCl, it could be HF. They're all going to do the same thing where they're going to break into hydrogen and then whatever this other molecule is. One's going to attack one side of the double bond, the other's going to attack the other side of the double bond. So, butene, 4 carbons, kind of the same situation as hexene. They didn't tell us where the double bond is, so I'm going to assume it's from 1 to 2. Now, I'm going to add HBr. So, hydrogen's going to attack this carbon. The heavier group, Br, is going to attack the first carbon. We're going to break that double bond. So then I get back, oops, Br, 1, 2, 3, 4. So the rest of the molecule stays the same. That hydrogen ended up here. So now I've changed it from an alkene to an alkane because I eliminated that double bond. And I'm sorry, my writing actually kind of looks like it's still there. Let me redo that. There we go. We eliminate that double bond. So butane, but now we've added a branch to it, Br. So 1 bromo butane. If, for instance, on the exam, it's like HCl or something, it'd be 1 chloro butane or 1 fluoro butane.
I have a question.
Yes.
So these two, it say 2 correct answers. On the final, it'd be 2 correct answers.
No, so on the final, there's 1 correct answer, and it's the lowest. So if you have it coming down to, 1 hexanol and, oops. So when I first wrote this, I was like, oh, there's 2 correct answers. And then I realized they want you to do Markovitch rule and that they've put both answers in the ABCD. So you have, so they're testing you on that, the Markovitch rule, which is to put the heavier group at the lowest carbon.
Okay, thank you.
I don't know, I started, I'm like,
Okay. So Kayla, you said you add a branch. When
Yes.
it's like chlorofluor or.
Yep.
Okay, chlorofluor. Oh, and you did that with the OH, so it would not.
Yeah, so the OH ended up being a functional group that we changed the ending here. Well, we changed the name in both of them, but we
Right.
changed the naming to a null because we added an OH.
Mhm.
As a null functional group. Whereas with the bromo, or say it's chloro, fluoro, it's still all single bonds, and we just end up, we can say the bromo, chloro, fluoro in the branch name.
Okay.
All right. Am I good to clear and move on?
Yes.
All right.
Yes.
All right. So now
No,
we're
that's alcohol. So
Yep. All right. So alcohol, right? We've identified our functional group. So it's going to end in anol.
Butanol.
So now we need to figure out how many carbons it has. We can go right to left, or I'm sorry, left to right or right to left. We're going to want to put our
Is P-E-N-T?
yeah, so we're going to want to put our functional group at the lowest possible. So we're going to end up choosing this numbering. And then like Angelica just said, pent. is for 5. So pent enol. What else do I have to tell my audience?
That is on the second. The OH is on the second.
Yep, perfect. So 2 pentanol. All right. Any questions? Okay. So you have one of these questions where, and it listed out exactly like this. You have, like we just identified in last, OH is a functional group and it ends in enol, or alcohol functional group ending in enol, but they have it sandwiched between, X amount of carbons. So the exception when you have 2 alcohol functional groups here is your ending becomes diol. Right? So we have 1, 2, 3.
Say that one more time. Sorry. You said that it becomes diol because of what again?
Because we have 2 alcohol groups.
Okay.
Mm-hmm.
So is that pentanol?
So it would be pent, and then don't take my spelling too literal. I think it's pent and diol. It's for sure. It's for sure
That's
got
the right
pent
one.
and okay. It's for sure got pent and diol. And then now this part seems a little silly to me because I think pent and diol, the di should imply that there's 2 functional groups sandwiching these, but we need to tell our audience that it's coming off of carbon 1 and carbon 5. All right.
Okay, I get this. It sounds because you're not counting the OH, you're counting.
Yep, counting carbons.
Oh, shoot, I thought I was on mute. I'm sorry.
So I have a question. Usually, like how we put the die in front or like the tripart in front when it's 2 or 3, we wouldn't that wouldn't be on one of the answer choices for this particular one, would it?
I don't think so.
Okay.
Do you care if I should have brought it up here with me? If I go grab it and so I can double check
Oh,
that.
go do what you got to do.
The nerves.
Angelica and Ken, did y'all finish all y'all aisles now?
Mhm.
Good.
Yep.
So it's, yeah, they're not gonna put a die in front of they're basically just testing you if you know what carbons they're coming off of and if you know to change the ending from enol to diol. Ooh,
Okay.
stairs just got me there. Okay. Any more questions about 10? All right. So looking at 11. So to name a structure, we first have to identify what functional group's present. What functional group is present?
Ketone.
Yeah. You guys are getting much better. At first I was a little worried. And so that takes on the ending anone. So now it's a matter of numbering it. We want to put the carbon or the functional group with the carbon on it at the lowest possible. So going from left to right here is more favorable than right to left. So we have pent again, and then anone because of the functional group. We have to tell our audience what carbon it's coming off of carbon 2. Additionally, we have to tell our audience about this functional group or branch coming off of carbon 3. So what kind of branch is that?
Methyl?
Oh, ethyl,
No.
3
It's
ethyl,
methyl.
It's a methyl.
methyl. What? Shit.
All right. So the full name 3-methyl-2-pentanone. All right. So I want to throw an example at you where let's say I still have a ketone group and I also have a branch. So if I number this right to left or left to, I've got them backwards, but no matter how I number it, my ketone group falls at 3. But the number of my branch does change. So in that case, what do we do?
The lowest, right?
It's still technically in the middle in 3, so wouldn't it still
Yeah.
be 2?
So yeah, so this isn't something we haven't really stressed, but say that I've got my my functional group at the lowest, no matter, say it's centered like this, and we have branches. The next move would be to choose the numbering scheme in which the branch is also at the lowest possible carbon. But only when we've prioritized this guy as being at 3 no matter what, basically. So if I saw something like this on the exam, I would go with this numbering scheme.
From right to left, right? Right to left.
Yes. So it would still
Okay.
be pentanone 3,
Mhm.
and then it'd be 2-methyl. In the example that I gave you, it we had to put it, we didn't want to do it the other way because it changed where that functional group was.
Okay, hold on. So which one is gonna be the correct one? The first one you did or the second? It's
They're both
the
correct.
second
They're
one?
just different. They're just different examples.
Oh, okay.
Mine is in a different, one is on 2 and the other is on 3. Okay, I see it.
Okay. Okay.
All right. So moving on to example 12. What functional group is present here?
Aldehyde.
Yes. So here's our aldehyde, right? Because it's on the terminal carbon. So it's going to take on the ending anal. So now we want to number our carbons, and we want to put this guy at the lowest possible. So I'm already going to start numbering from right to left. Right. So hex, because it's 6 carbons. Anal, because it's an aldehyde functional group. So here is the exception to, not necessarily the exception, but basically aldehydes are always going to fall at a terminal carbon. So they're always going to be at 1. So you never need to tell your audience that it's at 1. So a reminder, functional groups that work that way are aldehydes, carboxylic acids, they always fall at the first carbon, and amides. So basically, I don't want to say they're exception to the rule because they kind of fall under the implied 1 rule, but so if you only see
Under carbon.
if you only see hexanol on the the answer choice, don't freak out because it's correct. Not that yours is going to be hexanol, but if it doesn't have a number in front of it, basically, is what I was trying to say. All right. Questions? All right, moving along to 13 here. So this is one of the more complicated examples where we have the benzene ring. We have a double bond or 6-member with double bond ring and the functional group. Here we can simplify this as being all part of the parent chain here. So our 6-membered ring and the carboxylic acid group. And so in this case, it takes on the ending benzoic acid. And we don't need to give it necessarily a prefix. We just need to tell our audience about the branches. And so we can, the branch in which the carboxylic acid group is on is always going to be 1. Now I could go counterclockwise, horrible 3. But I would actually want to go clockwise as it puts those branches at lower carbons. So now I need to name these in alpha. So I have a chloro group coming off of carbon 2 and a methyl group coming off of carbon 4. In this example, you know, going in numerical order is the same as alphabetical order. So 2-chloro, 4-methyl benzoic acid. All right, so that's the answer to this one. Let's say, hypothetically, I switched these groups. So I would still do the numbering scheme clockwise because I want them to be at the lowest possible. But now my chloro group is on 4 and my methyl group's on 2. Be careful not to list it as 2-methyl 4-chloro. Make sure you follow the alphabet rule, which would say 4-chloro, 2-methyl, and I'm running out of room, so I'm going to jump the line down, benzoic acid. Okay, any questions about that? Okay.
So it will be alphabetical order for, like when we name them, just make sure it's in alphabetical order and then for the branches.
So basically what I'm trying to tell you is that the example that they give for you for this kind of problem throws that group, and it's not a chloro, but bromo and fluoro still are higher on the alphabet than methyl. So I'm just trying to give you a heads up.
Okay.
Because I see that they try to trick you. Okay. All right, so one of our favorites when we have the N. All right, so amide, and I spelled this wrong. It should have been amide. Whoops. But amide is what functional group? And this one's kind of easy because it's the name. right? So it's an amide.
Got a nitrogen on it.
Yep. So it looks like carbon double bonded to an O,
Oh,
bonded to an N with hydrogens, but the more bonds you put on the nitrogen, the more bonds you take away from the hydrogens. Besides the point. Okay. So bute, 4 carbons. So 1, 2, 3, 4. Oops. 1, 2, 3, 4. I guess I could have drawn it that way. I always like to put them on the right here. Okay. And then NH2. So we've tackled the bute and amide part. Now it's a matter of tackling the dimethyl off of NN. So NN means it's coming off of the nitrogen. And then the rest works the exact same as the other naming. So NN means we have 2 methyl groups, both coming off of the nitrogen. So I have a methyl group and then a methyl group. Now that takes away the bonds that could potentially have been to hydrogens.
So that took away the H altogether from the NH? Okay.
If it was just dimethyl, I'm sorry, just one methyl, then I would have left 1 hydrogen. And if there was no branches coming off of the N, then I would have left the 2 hydrogens.
Okay.
Questions?
So the first one is the final answer with just the N and the 2
Yep,
the 2 hydrogens, or
with the 2 methyl groups. All right. So looking at question 15. So I predict this is going to be one of the hardest questions on the exam. If you haven't watched my video lectures for, I think it's chapter 17, my YouTube video, you should definitely go watch that. So what happens here when you have this reaction and you're trying to determine what the product looks like? So basically, this nitrogen makes a bond to this oxygen. We drop all the hydrogens that are involved. And then we keep the rest of the molecules the same. Oh, I'm sorry. We attack this hydrogen, but the OH drops off too.
was that an NH2 or NH3?
It was an NH3, which I know is probably confusing since I just drew the other thing as an NH2. Okay, so then now we have 1, 2, 3. So we have to carry those carbons along. 1, 2, 3. So now it asks in the question for us to name it. It doesn't have answer choices as the pictures, but as names. So our parent chain here is the one with the carbon double bonded to a oxygen. So we have a bute and amide again. This time we have N to a propyl group. So N propyl, bute and amide.
Why did you use that side, the left side, to name it, and not the right?
Because we want to use the carbon chain that contains the functional group. So change this. So our functional group is here. And the carbon chain that contains that is the chain, the 1, 2, 3, 4 chain.
Okay.
Oh, no. Okay. So at the beginning of it, you had,
Mm-hmm.
oh, that was an oxygen?
Yes. So the reaction ends up dropping water. So the OH from here and then the H's
Oh, H.
here end up coming together and creating water. This picture that I pulled off the internet, I apologize. It should only have one N or two. It's got a lot going on. Just ignore it.
So 1, 2, 3, 4. So 4 and 3. So you still keep, okay. Pro, this is the key. The sandwich between the end, okay.
All right.
So can you go over that, just the part where you said, okay, when you put those together, you said both of the hydrogens drop on both sides to make the
Yeah.
N
So
at the end?
yeah, so I'm gonna clear this and just start over here. So the picture, I didn't realize this till just now. The picture I pulled off the internet doesn't have Crystal, your mic is still on. So the three, so your picture that you have on your exam would look like NH with something attached, and then this carboxylic acid with something attached. So what ends up happening is this group attacks this group, water comes off. So this oxygen, this hydrogen, and then this hydrogen come together to form water. And then the rest of the molecule merges. So if this is
Whoa.
1, 2, 3, 4, and let's call this A, B, C, we want to retain the 1, 2, 3, 4. Oops, need one more, 4. And then retain the A, B, C, D. And I'm calling them A, B, C, D because they are going to participate in the naming. The only carbons that participate in the naming are the ones that are directly, oops, I did number that wrong. Okay, eraser. I forgot to count that one. The only carbons that are going to participate in the naming are the chain that's directly connected to this group, basically. So then these carbons, yes, there are 3 carbons here, but this becomes a branch, a propyl branch. So to reiterate, this is N-propyl, bute, and amide. Because we've created an amide functional group, which is what I circled. Okay. All right. Okay. All right. So we've exited the organic of my practice exam. We've exited the organic. So there's what, 50 questions on the exam, and there's 23 that are organic. Just FYI, it is about 50% organic. All right. So now we're going into the biochemistry. So do we remember how to identify glycidic linkages? So your picture looks not exactly similar, but it's pretty easy to imply which is 1 and then which is 2. So if our first bond to this oxygen is going up, is it alpha or beta?
Beta,
If it's going up, it is,
beta,
yes,
beta.
yes.
Mic not working.
And then, all right, so then now we're looking at our second one. And if it's coming down, it's alpha, right? So beta 1, alpha 2. Now, if I were to stack these, let me just draw like a skeleton structure here. All right, let's say there's some stuff coming off. I just made that up. So if we're to stack it, this is our 1 and this is our 2. All right.
Alpha 1 or alpha then beta?
So then it would be, if it's coming down, this would be beta 1, alpha 2. Wait, no, if it's coming down, it's alpha. I'm sorry. You were right. You were right. Alpha 1, beta 2. Alpha 1, beta 2. Yeah. Okay. All right. Questions? All right. So, question 17. So, amino acids bind together and release water to create a peptide. This is through a dehydration reaction. What is the bond called that makes that peptide bond? I just gave it away. I'm sorry. So, peptide or amide bonds are what connect amino acids in a polypeptide chain. So again, this isn't the exact question that you're going to see on your exam, but to reiterate, amide and peptide bonds are the same, and they are what links amino acids. And this is a dehydration reaction. All right. So I'm going to be honest, this one I did take pretty much verbatim. I just changed what structure it was looking for. So the tertiary structure of a protein is maintained by A, a linear sequence of peptide bonds, B, hydrogen bonds that create alpha and beta sheets, C, hydrogen bonds, hydrophilic interactions, disulfide bonds, anionic bonds that create 3D shape, or D, several polypeptide chains and joining. So the correct answer here is C, tertiary structure, right? So when we talked about our line of students at the playground, we had our primary structure, which is just how they lined up one by one by one, right? So that's the linear sequence, right? And then we said, oh, Mary wants to hold hands with Johnny, but Johnny's 6 people back. And so now it starts to kind of snake, right? And we called those snakes either alpha or beta sheets. And that was our secondary structure, right? And so the holding hands was to represent hydrogen bonds. Tertiary structure was then when we start to become globular and 3D and we start to play Twister upon ourselves. And basically there was 4 different ways, bonds that made you become 3D and globular. And those were additional hydrogen bonds, hydrophobic interactions, disulfide bonds, and ionic bonds. And then we could say, you know, Miss Mary's class and Miss Susie's class came together and they formed a polypeptide, and that's quaternary structure. Okay. Any questions there? All right. So looking at 19, true or false, lock and key theory is when a protein changes its shape to accommodate its substrate in the active site.
True.
False.
False. Correct. So this is an example of induced fit. Right? So think about, I gave the example of giving a hug, right? You when you want to give somebody a hug, you put your arms out, and then once they get close to you, you end up changing shape to actually hug them. True or false? Enzymes release the activation energy required to perform, increase the activation energy required to perform a reaction.
False.
That is false. They decrease the activation energy. Additionally, though, this speeds up the reaction and increases reaction rate. So if you see this question on the exam, make sure that you are paying attention to when it's talking about increasing or decreasing reaction rate versus increasing or decreasing activation energy. Okay. True or false, RNA is double-stranded, uses the bases cytosine, adenine, thymine, and guanine.
False.
Correct. It's false. Why is it false?
It's AG.
Oh, go ahead.
Oh, go ahead.
So it uses U instead of
Yeah,
T, and it's single-stranded.
single-stranded.
Mm-hmm. All right. The next problem we would need our codon chart for. So I am going to pull up a codon chart in another tab here. I wonder if I can pull it to the side. We'll see if I can try to make it. Give me a second.
The codon chart is the circle chart, right?
Yep. Modified reference sheet. All right, so if
One thing I know is the first one is MET. I don't know nothing else after that.
You're right. Okay. I put this here. You can see it, right? And then we can also kind of see this. Okay. Yeah, we can see it. Okay. This is nice. Okay. So I'm going to divide it into thirds. Not thirds. I said this when I lectured too. Into three codon pieces, right?
Mm-hmm.
So A, U, G, just like Kenny had said, met. U, A, C, tyrosine,
Mm-hmm.
and then A,
Right.
and then G, U, vowel, and then G, vowel again, is that? Yeah, that's right. Okay. Oh crap. Okay. There's a undo too. There we go. Okay. And then finally,
Read it, man.
A, which is a stop codon. All right. So if you remember when we were talking about this in class, I was like I was double checking your exam because I was like, wait a second. So your exam does the example they give you does end in a stop codon, but they don't the answer choice that's correct, they don't explicitly write out the stop codon. So I'm gonna have to make an edit to this one because it's kind of confusing because then the other answer choices, they all end in stop codon. So it's like, why did you do that? But yeah, I'll make that edit when on exam day. I'll tell you, what to look for. Okay. Any questions about that one?
Yeah, is that chart gonna be available or we
Yes.
gotta know? Okay.
Yep. No, please. Oh my gosh. Do not. How much panic would I instill in you guys if I said you have to memorize? So here's the sheet I plan on giving you. So you'll have, let me clear all this stuff so it's not. Okay, so you'll have this chart, this, and then I'm also, the backside will be the periodic table. I don't really think that you're gonna need it, but I just thought might as well, I guess.
Okay, thank God. All right.
Yep. So so the one thing that people were wondering is if you get the chart of functional groups and what they look like, and that answer is no. I want you guys to be able to identify them. From the people who are in this group, you seem to be good at identifying. There are some people that need to work on it, but they're not the people that are here, ironically. All right, moving on. All right, so 23. A polypeptide chain is broken down into individual amino acids to be digested and utilized by the body. This is an example of anabolism or catabolism.
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 Yes. Right? So because we are breaking something down. If I said the opposite, I said, oh, our body was building amino acids into a peptide chain. That would be anabolism, building up. Okay. All right. So now we're moving into the questions from the cellular respiration part of lecture. And I will be honest, I basically gave you kind of verbatim, except for they're listed as, A, B, C, or D. So what is the final product of glycolysis? So
Pyruvic acid.
yes.
ATP.
Yep. So pyruvate. So ATP is there, yes. But it's not the primary product. It's just kind of a byproduct. So there are specifically 2 pyruvates, but that's not something they really stress on the exam. All right.
So is it gonna say pyruvate or like pyruvic acid?
It says pyruvate.
Okay.
But actually, I also tricked you. I added this one, but that one's not actually on the exam. This one is. These 3 are, or 4. Don't stress too much about that one, but yeah, no. All right, what's the starting product of glycolysis?
Glucose.
Glucose.
Yes. All right, break down those sugars. What's the starting product of the Krebs cycle?
Acetyl
Câu A.
Yeah, co-A.
Yes, perfect. FADH2 and NADH are produced by the citric acid cycle. Why?
They're transporters.
Yes. So they are electron transporters. What did I say? And we transport them to the final step. And what is the You know what? That's actually all you need to know. That the main purpose. Okay, the question is, what's the main purpose of the citric acid cycle? To produce those transporters. All right. True or false? FADH2 and NADH are taken to the electron transport train and transported across a gradient in the final step of cellular respiration.
True.
True. Yes. Okay, so all right, yeah, we covered them. I lied. Okay, I'm sorry. I did lie. This one is there. It's just a lot wordier than I thought it was. Okay. It combines these two. It asks, what's the purpose of a glycolysis? To break down glucose 2-pyruvate. That's why I wrote it that way. Okay. All right. Questions there. All right, so we'll clear this. And then it looks like the final question I had for you guys on the practice exam was, what is the difference between glycogenesis and gluconeogenesis?
I put
I think-
glycogenesis. You get glucose from carbs, where in the other one, you get the glucose from non-carbs. Is that right?
It might be, it's not necessarily what I was looking for. So
Okay.
glucose is the usable form, right? This is like in our blood. Glycogen is our storage form. This is housed in our liver. So glycogenesis is the process in taking glucose to glycogen. Gluconeogenesis is the opposite. And this also means the same thing. I think in your lecture slides, they were gluconeolysis. Same thing as I can't spell. Gluconeogenesis. Okay. Do you have any other questions? I was thinking I could pull up let's see here, modified. Give me a second to stop sharing my screen. Modified step.
I know I missed it, but what was the answer for number 16?
16. Beta 1, alpha 2.
Oh, okay, I got it right. Okay. Make sure I understand that.
All right, so actually, let me pull it up. This is what I want. Okay. So in the last page of the answer key that I had posted the study guide here and what I'm gonna need to grab my charger here in a second here. And you know, like what if it was covered. So we did not cover what an alkane is. What is an alkane?
Okay.
Okay, so I didn't have a practice exam question for that, but alkanes are where all the carbons are single bonded, right? We talked about being able to name an alkane. We talked about identifying an alkane from its structure. We talked about determining which one has the highest boiling point. We talked about functional groups, and I pointed out that you have 2 questions about functional groups. We talked about alkenes, all that, cis and trans, reactions, being able to name an alcohol, and we had 2 of them, right? We had that diol one. All right, so 5. Okay, so here's one we didn't talk about, but I did cover it in our last lecture. Do we remember what the answer to this? So there's a test that distinguishes aldehydes from ketones. What's it called? Anybody remember?
Well, it's not one of those tests we did in.
No,
In
it's
lab, right?
ironically not a test we did in lab.
Mhm.
Hold on, say that, ask the question again. I'm sorry, I didn't hear it.
So what is the test that distinguishes aldehydes from ketones?
And that's not talons.
It is.
Tolins.
Tolins,
Not
Reed.
talons.
Yeah.
Sorry.
Oh, it's totally rich. I was gonna say the ketone in the middle and aldehyde. Okay.
Oh, yeah,
I
no.
was gonna
I was
say
saying it,
that.
but I was on mute.
All right.
Alright.
So we covered this today. Okay, so this is something we didn't necessarily cover in the in this review session, but you know, what is saponification? Let me see if I can find that question.
We just went over that last I forgot who said that was.
So if I said that I had some kind of acid, right, and I mixed it with a strong base, specifically NaOH, right, that is saponification. So an acid, right, usually a fatty acid, but look for acid in the name plus NaOH. That's a saponification reaction. Additionally, we wanted to know which you have a question that's which lipids are non-saponable, right? And I had this also in your lab. There was 2. Do we remember?
You said lipids nonsense.
So there's prostaglandins. They are unable to go
And
undergo saponification and
steroids, right?
yes, steroids.
You don't want to know what I was gonna say. I was about to say carbohydrates.
Well, I
Let
mean,
me be quiet.
that is kind of true, I guess, because this is all about lipids. You know? All right. So we talked about this. We talked about this. Okay, so what about this one? Neurotransmitters are commonly what functional group? And this was something I said in the last lecture. Amines? So amines, so things like what's a common neurotransmitter? I don't really know. Serotonin, right? It's made up of amines.
So that means, okay.
Mm-hmm.
Means.
All right, I'm gonna run and get my charger before bad things happen. So I'm gonna clear this. Looking at stuff from module 6. So you do have to identify a common disaccharide, and I promise you it will be stupid easy. All right, we talked about this. All right, so know the properties of common the properties of sucrose, basically. And I asked you this in your lab. Do you remember what's so important about sucrose? So sucrose is a sugar that cannot be
I was gonna
reduced.
say-
Yep.
I was gonna say something with sugar, but I was like, no, the last couple of times I've been getting it wrong, so I'm gonna be quiet.
Right. So that glycidic linkage is unable to be reduced. So it can't go from a disaccharide to a monomer, right? Prevents the open chain. Right. All right. It looks like I added this later. Okay, so know the difference between glycogen and cellulose, and I ended up actually deleting that question, so don't freak out too much about that. Okay, so understand biological membranes and how they are composed. So you have 2 questions, right? And we covered this in the lab. So I believe I ended up actually deleting one of them because I found an error. But I did leave one. So let me find the other ones. All right, so what lipid mainly makes up the cell membrane? You guys know this. They have polar heads and they have non-polar tails.
Hydrophilic or hydrophobic?
A phospholipid.
Phospholipid, yes. So phospholipids make up majority of the cell membrane. All right. Oh, we already covered this. Know which lipids can undergo saponification and which can't. All right. Okay. Here's one we didn't cover. Understand the properties of common lipids, specifically sphingomyelin and phosphoglycerol. Or glycerol. Okay, so what's the question asked? How do sphingomyelin differ from phosphoglycerides? So I think I had one slide on this it was in the lecture, which we had to move rooms. So Sphingomyelin has an amide bond. And I think, yeah, if you remember that, you can get yourself through the rest. All right. So be able to describe the components of an amino acid. So we talked about question 17 on the practice exam, which was the peptide bond, what kind of bond. And then also you'll have a question, which I put, it was just like question 1 in the lab 6 discussion, right? So what is an amino acid composed of?
Proteins.
Proteins are composed of amino acids, but specifically a amino acid is a carboxylic.
Monomer and polymers. Well, a
So
polymer chain.
Yes, both of those are right. But what I'm looking for is the
Huh.
3 parts of an amino acid, which is a carboxyl group, amino group,
And a sugar group, right?
and then some R group. All right, so let's reiterate that. 3 components. Carboxyl group, which we called carboxylic acids, means the same thing. They have the terminology as carboxyl group. Amino group, which we called amines, but same thing. And then an R group. Okay, we cover that in question. Okay, so this one, so you had a question on your midterm that talks about reaction rates. The things that could affect reaction rates were like temperature, pH, and a catalyst, right? And I think the question was like, which ones affect reaction rates? And then the answer was all of the above. You have a similar question, but it talks about what can affect the rates of enzymes. And it's literally very similar, right? So pH can affect enzyme activity, temperature can affect enzyme activity, and this time instead of catalyst, because enzymes are catalysts, we can affect the enzyme activity with substrate concentration. So pH, temperature, and substrate concentration. All right. For that, okay, so a common irreversible inhibitor. I talked when we talked about irreversible inhibitors, I think I only gave you one example because that was the one that I wanted you to remember. Anybody happen to remember by chance? I said it was also a polyatomic ion. Cyanide.
Oh, the cyanide.
Yep. CN. All right. Before I clear this and move on, do you want me to hold this for a second?
Yes. Okay, thank you.
Anybody else? Okay. All right, so module 7. Okay, so we didn't cover this one. Be able to define what translation is and what it does. You guys tell me, what does translation?
It takes RNA to
Protein? Right, so mRNA is taken to the ribosomes.
Oh,
right? To assemble the polypeptide, which is a protein, right?
The protein, right?
Yep. Nothing to do with DNA. DNA is transcription. All right. Why are vitamins important? So they serve as precursors to cofactors. Precursors to cofactors, right? Do we even remember what cofactors were? They were helpers for enzymes. All right, so we covered that one. So the difference between trans fats and unsaturated fats, right? So trans fats, they have a kink in them, right? And they had that kink, and they were solids at room temp. Unsaturated fats. Oh, I'm sorry. They're wait. Oh, I don't like this question. Okay, so the question is choose the one which is not true. And they're wordy as heck. Okay, so cis and trans create this kink. Right? Cis are liquids at room temp. But trans are solids. So I will say this is the question's a select which one is not true, and the things are wordy as heck. I would focus on this, looking at this part of the problem. Okay, so we covered all the cellular respiration stuff. We defined glucogenesis. And then, all right, so be able to define beta oxidation. Remember what beta oxidation was? It was the breakdown of fatty acids. And then finally, something else we didn't cover, but hopefully you guys remember. So if amino acids can't be synthesized, are they essential or non-essential?
non-essential. If they can't be,
They're essential.
essential if they can't be.
If they can't be synthesized, they're essential that they're in our diet because we have to eat them because our body can't make them. It's like kind of backwards. All right. Destiny, I know you just joined. I have good news for you. I'm recording the lecture and sending it out, or not lecture, but review session, and sending it to those who have attended so you can see the stuff that you missed. But I do think we're kind of wrapping up here. So any other additional questions? Now's the chance. I'll take questions on OWL questions.
I have an out question.
Okay.
Do you want me to show
Yeah,
my screen?
let me just so let's just make sure any more questions about exam review stuff, and then we'll take out questions. I guess I should have said
One
that.
more question. Will you be uploading this sheet as well?
Yes,
This
this is actually already uploaded in the announcement titled.
No, like I'm saying like with question 21, question 22, like this one.
Yeah, this is in the key. This is on, oh, let me clear this. So this announcement that was posted on April 9th, it's in this document.
No, I have the sheet. I'm saying like question, like you have it on there, like question 21, question 1,
Yes.
question 5, question.
So that's at the end of that.
Oh, okay, okay.
Yeah.
Okay, that's the sheet I don't have. See, I printed the other one out. Okay.
Yeah.
Okay, thank you.
Yep. Okay, any other questions about exam review specifically stuff? All right, so now Alexis, let me stop sharing my screen and then give you the access. Host. Okay.
Okay. I don't know what I'm doing wrong right here.
Let me pull this up. All right. It's probably something stupid. Do you want to try to just attempt your other question there?
Oh, it's the same one.
Oh, okay. Okay, then let me look specifically. Okay, then thymine structure. Which thing? Oh, I see it already. Okay, so these ends need to actually be in the ring itself.
Okay, so look, I did one like that and it still marked it wrong. So I'm gonna try it on the other one
Yeah,
so
no,
you can see.
no, they're like in it. Like I wish
Look,
I
I'm about
Yeah.
to show you. So
Yeah, you gotta click
boom.
on the end and then on the ring.
Right. So I had one like that as well, and it was marked wrong.
Well, maybe it was a different base though.
Sorry, y'all, I gotta look.
That's fine. Okay, so now when you go to put your nitrogen, put them over those carbons. Yeah, like this.
Okay, now
Okay, now
see.
it should be good. Yeah.
maybe it was a different base. Okay, so maybe it was, and I did. It was thymine though. I don't know. Maybe it was from RNA, and I didn't notice that or something.
Okay.
I don't know, but thank you.
Yeah.
And let
Anybody else?
All right, thanks, Professor Daniels. See y'all
Thank
Tuesday.
you.
Thank you.
Good luck on your guys' exam. You guys got this. Email me if you have any other questions.
I got more questions about Al, but I was trying to get somebody else.
Okay. Maybe you're the only one. Go ahead then.
Okay, let me go back to my screen.
Actually, I am gonna ask something.
You have a question, Ken?
I'm just looking at, I was looking at my owls. For some reason, I'm trying to name it, but it's not, the names ain't coming out right.
Okay, here you can help him first.
Okay. Let me. Okay.
Trying to find my sheet.
Lexus, I got the same one as you, right? For the owl?
Yeah, the thymine.
No, the one with the,
That she was just about to pull up or something.
yeah, that you were about to pull up.
Oh, probably so.
I don't know if I.
Ooh,
Oh, shoot.
is that right?
You working on sharing your screen there, Ken?
No.
Oh,
Okay.
Jesus.
I was looking at my screen. There.
Oh, no, I turned it in. I fixed it. Okay.
Oh yeah, Crystal. I noticed today you had all your stuff. 100%.
Yeah.
You'll be getting 100% in the owl category. Woo.
We're here.
Thank you, Jesus. Oh, I did have one that, remember you had given us 17 and mine didn't come off. Did you, I think that was like mod 4.
Yeah, I would have changed it maybe around 3 today. Did you notice? Have you looked in there?
No.
Okay, well, I know I for sure am like in my you're getting 100% on my end.
Okay.
So here's the thing. Owl might like try to resync overnight, but like what I'm gonna do is like send you guys after you take the final, and if you've completed all your owls, I'll send you an email like, all right, here's what I have for owl assignment 1, 2, 3, 4, 5. That
Okay.
equals this percentage. You know, that's what I'm basing your final grade off of. If owl tries to resync in the middle of the night, like please understand that this is the grade that I'm putting in. So
Okay.
hopefully that will clear up any discrepancies.
Okay.
Because I know some people right now, it keeps resyncing and it makes it look like they have a higher grade than they do.
Oh.
Or sometimes
Yeah, because mine gave me 100%, and I know like 2 of the owls I'm gonna have points taken off.
yeah, so yeah, it's I hate owl. I hate it. I hate it. I hate it.
Yeah, owl sucks. But
All right, so what are we doing here?
Do you have an easier way for me to remember functional groups? I didn't wrote them down. I still can't memorize them.
I think I just wrote them down over and over and like quizzed myself and like kind of played. You could, oh, you know, it'd be kind of cool. You know that game Memory where you like you have
over.
yeah, you flip them over.
Yeah.
Try to like you could make like on a post-it note, you could have OH and then on another post-it note alcohol, and you could try to like match them to each other.
Match them, like the matching game.
Yeah, that'd be
I
fun.
can do that.
Maybe that'll help.
Yeah, look, right now I'll try anything. I mean, I'm familiar with it when people talk about it. Then I'm
Yeah.
like, oh yeah, that's what that is.
Yeah, but just yeah.
Mhm.
All right.
All right, mine's is simple. I mean, it should be. Share.
All right, I'll see you guys on Tuesday.
Yeah. All right.
That. Okay.
All
Please.
right. We can do this. All right. So, okay. So these are kind of the similar to the alcohol where it's got like we have to add di in front of it. So it's an alkene, right? But it has 2 alkenes. So we want to put these at the lowest possible we can. So 1, 2, 3, 4, 5, 6, 7. So because it has 2 alkenes, it's going to take on the ending diene.
Right.
So it's heptadiene, and I might be spelling that wrong. Let me like Google that. Nope, I spelled it right. Okay. So then we need to tell our audience where those are.
Right.
So it spans, yeah, don't type it in quite yet. It spans 2 to 3 and then 4 to 5. So we just tell them the lower of the numbers. So 2 and 4.
Right. Yeah.
And then we have to tell them about the branch here. So there's a branch coming off of
6.
carbon 6, and it's a methyl.
It's methyl because it's only 1.
Yep.
Okay. So if there was like a CH2 here, it wouldn't be methyl, it'd be ethyl then, right?
Not a CH2, but rather 2 carbons in a chain.
Okay. Okay.
But also, yeah, kind of correct if you did. Yeah.
I mean,
All right,
carbons. I mean, yeah. Okay.
go ahead and type in this name.
Go here. 6. That's me.
Yeah, that's weird.
It does that. I'm on 4. 6-methyl. 2-carnosyl.
Looks-
Dean,
Good.
looks right.
Yep.
Okay.
Woo. All right, you think you can do that on another one similar?
Like this one. Okay.
Yep.
So we still got 1, 2, 1. See, this one I'm mixing up because I want to go 1, 2, but I know this is separate.
No, you're right. You're right. So 1, 2, 3, 4, 5. You're counting your parent chain, right?
Yes.
Yep.
Okay. So 5 would be pent. So
Yep.
I know I'd have a pent there,
Yep.
something in front of it. All right. So then I got 2. So if I have 2 double bonds,
Yes.
That's going to give me a ethyl then
Dying.
diene. Okay. That
Now,
part.
and
So
we have to tell the audience where they are at. So they span 1 to 2 and 3 to 4, but we only have to tell them the lower numbers. So 1 and 3.
right.
And then what else do we have to take care of?
You got to take care of this right here.
Yep. So that's 2 methyl 1,3 pentadiene.
2 dash 1
So don't forget a hyphen between the 3 and the key there.
two methyl dash 1 dash 3 pentadiene. There you go. There you go. Okay. Okay. Thank you.
Yes, no problem. All right, we got about 10 minutes left. Alexis, did we want to come back to your problem?
Yes, we can.
So Ken, I'm gonna stop sharing your screen, okay?
I'll cut mine off. That's cool. Thank you.
Yep.
Okay. So I have
Yes.
this. I don't remember how to determine
So
the 3-letter amino acid.
yes, that's using the codon chart, which you'll get on your exam. That's the circular thing.
Oh, then that'll be easy.
Okay.
Okay. Never mind. I can do that on my own.
All right.
All right, here we go. I just don't know. I just don't know.
I don't know either. All right, open up another question.
Well, here go. Okay, here go one.
Okay, a protein that contains all of the essential amino acids, it's called I don't know.
Me either.
Destiny or Ken, by chance, did you get this question? And
Let me see. Was protein that contains the essential amino acids. Wait, number 7? Hold on. Let me pull back. I think I did.
Number 8. Yeah, module 7. Number 8 on module 7. And then I have this one too, which is the following macronutrients are digest the most slowly. Wouldn't it be
I think it's carbs.
carbs? Yeah.
I
didn't try this one.
I thought it was lipids that are digested slower than carbs.
Oh yeah, you was right.
All right, I think when I Google it, I get complete proteins, but double check with if somebody else also had this. Ken, did you?
I'm not gonna submit it.
Can Google it.
And then see, like I feel like this is just too much words, and they gonna mark it wrong because it's too many words.
A protein that contains all of the essential acids. It's called I think that is right. It's
Okay.
memory.
Oh,
Okay.
Whew, that was tough. Okay, what's the name of the chart I need for this one? So I can.
Just type in genetic code, see if it, there's a lot of them.
Okay, all right, I'm gonna work
Yeah.
on. I can do that one.
All right.
Oh yeah, one more question. Because I came on late. I know you said you got it recorded. So
Yeah.
for number 17 on
Okay.
the practice exam and number 18, I put question marks next to them because I wasn't sure if I was understanding the question right, or maybe I just didn't know the answer.
Okay, let me pull it back up. Because I off the top of my head, I don't know what's question 17 and which one is it?
Oh, here, I got it right here if you want it.
Oh, yeah. Thank you. That works.
15. Okay, right here.
Okay, so 17, 18 is what you're asking about?
Yes.
Okay, 17, yes, that's correct.
Oh, okay. And then
18
18
is C.
Okay, that's what I got on my paper copy. I put C or B. I just wasn't sure. Okay, so it's C. Okay. Okay, I think that is it.
Awesome.
All right. Thank
Wait.
you so much.
When you look at mine, I'm trying to walk through it, right? But I think I'm doing it right. Naming the extras is what's getting me all flipped around.
Okay,
Let me
share
see.
your screen again.
Okay. Did that work? I think I did it.
No, I can't see it.
One person share. I clicked the green. Oh, never mind. There you go. Blue button. Duh. Okay. So
Okay,
I got I'm on the second method. So I'm
yes.
methyl for this one. It's a 5,
Yes.
it's a pent, so I
Yes.
wrote it already. 2 methyl, and then this would be di.
No, that would just be ethyl. So the
I thought it'd be diethyl. That's why.
no, it'd be diethyl if it was one methyl group coming this way and one methyl group coming this way.
Ah,
But
okay.
because they're directly connected to each other, it's just an ethyl.
Okay. So 2 methyl.
3 ethyl.
KYL.
Pentane.
Right.
They're all single bonded carbons.
Okay. So I knew how to get my parent.
Yeah,
Kinda, that second one is what I kept getting wrong.
yeah. Oh, wait, what? What is it?
Here, we'll start that over.
I don't know what it doesn't like about that one. Okay, I apologize.
That one. That's a triangle.
Oh, we can do it, but
And
okay,
then
let's start with this one. Okay,
7. So it's 7 carbs.
yeah,
So I know it's hept. So I write that. AC.
yes.
And
Hept what?
it's a A&E, ain't it?
Yep.
And they're all single bonds.
Yep.
Heptane.
All right,
But-
so then now we need to take care of that branch.
So it's going to be 4 no matter which side I come from.
Correct.
So 4 would be, is it still a butt?
No, so here
It's in the middle. So it's an alkane. I mean, it's a ketone.
yeah, so the branches are just based off how many carbons are coming off of it. So
Okay.
how many carbons are coming off of it?
Just one.
So it's a methyl group
Okay.
coming off of carbon 4.
Oh.
4 methyl heptane.
So I'm thinking too much.
You were You were choo, 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 choo.
And that's it.
Yes, if it counts us wrong, I'm gonna be very confused. Okay, we like that.
So then,
We could show me
Yeah.
that first one again. I mean, we could do it, but I don't really want to. This one's not bad. This one will be good. Okay, so our parent here,
3.
1, 2, 3, 4. Or I'm sorry, 1, 2, 3.
But so this is die because they're up and down right through the
Yep.
middle.
Perfect. Perfect.
So I got 3, so it's a prop.
Yep.
A and E, because it's
Yep.
all single bond.
Yep.
And then I know it's a die, but so it's still a methyl though. So it's
Yes.
it's a 2-dimethyl something.
Yep. 2,2-dimethyl propane. You have a B right now.
Oh, because it's 2-2 because one up top, one up bottom. the
Yep.
second two. And we're gonna say what it is by calling it the
Di. Yep.
DITHYL.
Okay, but make sure.
Propane.
Change that there.
Yeah. Get over. PIOPAN.
Nope. Too many P's.
Please.
There we go. All right. Should like us.
Yes.
Woo.
Okay. I don't think
All
I
right.
got it. Oh, 1. Is that all I got left? I think that's it. No, just this one. Oh no, I got more. I ain't know. So
All right, we'll do this question. Well, first I want to see Destiny's still hanging out, so I want to see if she has a question. Or if she's just hanging out, listening.
I'm just hanging out listening.
Okay. All right, Ken, we'll do this. We'll get you to master this category and then I'll call it since it's 8 o'clock, okay?
okay, thank you.
Yeah. So do you want to start with this one?
I
Doesn't
mean, I think they're all probably about to.
Yeah, doesn't matter. Okay.
Mm.
So you can manipulate that however you see fit.
So, I mean, I see 3 carbons right here.
Yes, so this
do
one's a little tricky. We want to make sure this double bond is in our parent chain. So 1, 2, 3, 4. And then we'll
Oh,
consider this group a branch.
okay.
So
Okay.
so if it has 4, what's the like parent chain
That's
name?
a BUT, but
And then it has a double bond, so E-N-E, A-N-E, Y-N-E,
ENE.
E-N-E.
Yeah.
Have to tell our audience that it's at the first carbon. And then we have to tell our audience about that methyl group too, right?
Right, which is gonna be on the second
Yep.
2-methyl.
Yes. So I think it's gonna want 2-methyl/1/butene. It might also take, if we get the little air, then it's probably looking for this, in which we omit the 1, but let's see what it likes.
What is that? It keeps doing that.
Yeah, I bet you have a virus.
I wouldn't. It seems like every time I'll touch my trackpad with like the palm of my hand and it does that.
Oh, then okay, then that's probably some kind of movement that is like, okay. It's funny. Whenever I get pops up, I'm like, oh, I must have a virus.
I don't do anything on this computer besides homework
Oh.
now.
Oh, then yeah, that's unlikely.
2-methyl 1-butene. Oh, my nose is killing me. Okay.
There, okay, it does like that. Okay.
I wonder. Hold on. I want this. I like. Okay.
Okay. So, parent chain, what do you see?
1, 2, 3, 4. I see 5.
I would agree. I would have counted, yeah, I want to count them this way though, but yeah, I agree.
Oops, well, I counted from the other way, but I guess it did make a difference because we're still CHC, we still got the 2 on both sides of double bond. And
No,
right?
it would matter. So here it puts it at 2, where there it would have put it at from 3 to 4.
Okay.
So I see, so 5 is what?
5 is my just a regular carbon.
Pent.
Oh, yeah, yeah. I'm
Yeah.
sorry. Pentene, yes.
Yep. We have to tell our audience it's coming off of 2, and then we
Yes.
have to tell our audience about those branches.
Which are coming off of 3 and 4.
Yep.
So
So since they're both methyl groups, we again can say dimethyl, but we
Uh-huh.
just, instead of saying the same carbon like 3,3, we now say 3,4
4.
dimethyl.
Okay. Slash. There.
Nice.
See, I can do it when I'm sitting here talking to you. I don't know what it is when I'm sitting here not.
Yeah. Guess it just takes repetition.
Mm-hmm.
All right.
I'm looking at your chart too, so that makes it easy too.
Okay, good, good.
Thank you.
Yes, thank you. All right. Have a nice night, guys.
You too.